integrated pre-breeding program for wheat and barley. So I thought today I'll just give you a quick overview of how we're going about to do that. So I might start with the current paradigm for pre-breeding that's in Australia at the moment for wheat and barley. So initially we start with a discovery phase when we're working with any kind of trait. So traditionally what's done is you take a resistant line and a susceptible line, you make the cross, you produce a mapping population. So for wheat it's an inbred species, so this involves many generations until that population is completely inbred. And that gives you a population which you genotype and then you phenotype for that particular trait of interest. So whether it be disease resistance or staggering, whatever. The problem with this is that it takes a long time. It can take 5 or 6 years by the time you produce the population and collect enough data to validate this gene discovery that you've made. But also, it's a biallelic system. There's only 2 alleles. You've only got a very limited germplasm pool that you're looking at. You're only looking at 1 cross. And in reality, what breeders are looking at is this huge array of genetic diversity that they're trying to work with. So it's not ideal. And so what normally is the second phase is this validation step. So once you've discovered your gene in this little population here, the next step is to rapidly transfer this gene, I guess, we'll try to do it quickly, in a number of different genetic backgrounds or different varieties for breeding companies to then make crosses in their program, which is another cycle of crossing away. So this stage might take another 5 or 6 years, but then to get that gene or trait into a new variety might take another cycle. So we're talking, many years, and if not ever, some of these traits have been transferred into varieties for farmers to use. So at the end of the day, we're trying to deliver these things. But it's not just about a single trait. In reality, this process it has a single trait focus at the moment. So researcher A is working on rust, researcher B is working on root architecture, crown rot, etc. This is happening in parallel with no integration whatsoever. So you can see that if you wanted to combine some of these things, how it becomes a very challenging job for breeders to do. It's essentially it's a gene numbers game. Each of these traits, for instance, it might be controlled by any number of genes. So you can see if you pick these 3 traits, rust resistance, root architecture, Jack just explained how that's a complex trait, 5 or 6 genes. Crown rot resistance, another complex trait that has eluded breeders so far in varieties. And so if we want to combine these things, we're talking maybe 20 genes. And so if you were to create a double haploid population in your breeding program from 2 parents, which is then the progeny is segregating for these traits, and you wanted to combine these 20 genes, you would have to evaluate 1 million double haploid plants for these traits. And that's really unfeasible to be evaluating those sort of numbers phenotypically in a breeding program. So we're talking quite a number of breeding cycles. And in reality, plant breeders are really dealing with many more than just 20 genes. So they're not just interested in these 3 traits, as you know. So just in summary, the current paradigm is slow, inefficient, expensive, and somewhat ineffective because many of these traits have not reached the farmers. So what we're talking about is developing a more of an integrated pre-breeding program where we have integrated effort across organisations and traits which are working on these different traits. So, but not only that, but we're looking to have an integration with the validation step. So if we can simultaneously discover these genes and validate them at the same time, hey, we cut out a whole step in a whole maybe 5-6 years. So we're speeding up the process. And so in doing this, remember back to the gene number problem, so we think that this extra phase in here, where we're packaging traits together in the combinations that are needed for breeders, will actually speed up the process at which these traits actually enter varieties for farmers in the field. So we're proposing a new paradigm in this integrated sort of approach, which is faster and more efficient, and hopefully in that it's less expensive too. So to give you sort of an overview of the phases of the platform, we're essentially using this nested association mapping platform as the discovery phase in our program. It was originally developed in maize in the US. It essentially involves a reference variety or multiple reference varieties, which are preferred varieties for the region of interest. So in the northern region, we've chosen Suntop and Spitfire wheat varieties that are preferred. And so it involves then crossing many donor lines or exotics that have diversity for the traits that you're interested in or your researchers are interested in. And so crossing them to the reference varieties, and as a result, you end up with a small number of lines, I guess, is preferred from each of those different crosses, rather than many lines from one single cross. So you have this nested structure. You have the advantage that you are incorporating historic recombination. So the differences between those donor lines that are going into the population, as well as recombination that's occurring when you're generating the population. And so what this really does is take advantage of the high throughput genotyping technology that we have at our fingertips now. Really, whereas the old paradigm really wasn't using this very well. So what it allows us to do is to, first of all, map these genes very precisely with that power, but also it allows us to examine them within a relevant context, because during the development of this population, we actually apply selection in the field in the F2 generation, and that selection is for an appropriate plant height and maturity. So we're not looking at these new genes in things that are 2 meters tall or extremely late, so they're within an appropriate context. So it means a lot more, and it means a lot more to the breeders. We're developing useful markers, and so we're also, one advantage is that when you find a gene in this particular subpopulation of your NAM resource, you can look across the resource and you can look at its frequency in those other subpopulations and its interaction with other genes controlling these traits, and also pick up negative associations with other traits much earlier. So rather than the breeders or the breeding companies picking these things up, you might have a new gene for rust resistance, for instance, and it's actually associated with poor quality. Well, we'll pick that up in this sort of resource where normally you wouldn't be able to. So that's where we're kind of up to at the moment, is actually we've just finished developing the phase 1 NAM population. Really exciting stuff. So it's 4 reference varieties from about 25 or so crosses, and so it consists of about 1,300 lines. So a really powerful resource for our group to be working on to dissect these genetics of these complex traits. Now, in terms of the packaging, once we've dissected the genetics and once we have good DNA markers for these traits, in terms of packaging them together, we have this excellent tool called speed breeding that we developed over the last 7 years at UQ. Now, this involves growing the plants under constant light and temperature to accelerate plant development. We can now achieve 7 generations of wheat per year in this system. That means we can go really fast, but there's no real advantage in going really fast unless you've got a way to select for these traits. So we've developed high throughput phenotypic screening methods for particular target traits we're interested in. That includes things like grain dormancy for pre-harvest sprouting, yellow spot resistance, adult plant resistance to rust pathogens, crown rot. And we've just recently, with Jack and I, we have a PhD student who's developed a system that allows us to screen root angle in wheat, and not only just evaluate root angle, but actually screen and evaluate 1,000 wheat lines in the space of 1 week. So we think that, we're talking about what's the next, what's going to take us to the next level, and I say that you have to look out for the root revolution. It's coming, and also the power in combining these traits together using the technology that we have at our fingertips now can really take us to that new level, hopefully. So Of course, we'll be using the information generated from our mapping and understanding the genetics using these GBS marker platforms in a form of marker-assisted recurrent selection. And so at the end of the day, these are the sort of outcomes we're delivering to the companies who are commercializing the germplasm. So we're developing more elite germplasm, better adapted genetic backgrounds, so new genes, marker trait associations, understanding how these traits work genetically, and these trait packages which really shortcut the time and the number of breeding cycles that are needed to get these combinations in the field. So for instance, if you look at how many wheat varieties that are suited for the northern region, how many are resistant to crown rot, resistant to yellow spot, there's not very many. That's it.
That's it.
Yep.